Welcome to Meet Them. Welcome to biology class of Standard 11 NCRT CBSE syllabus. So today we are going to continue with chapter 2, that is biological classification. We will be seeing part 2 today. Before we go to part 2, let us revise part 1. So what did we learn in part 1? That we actually learned about the 5 kingdom classification, but how was it evolved? So to begin with, Aristotle, who made the most scientific classification on the basis of simple morphological characteristics of the plants, and he classified plants into trees, shrubs, and herbs. Then Linnaeus came up with 2 kingdom classification. So at the time of Linnaeus, this classification was established, which only consisted of kingdom Animalia and kingdom plants, but it did not differentiate between unicellular and multicellular organisms. It did not differentiate between eukaryotes and prokaryotes. It also did not differentiate between the photosynthetic blue-green algae and non-photosynthetic fungi, and so on and so forth. So it had many flaws. Thus, in 1969, it was R.H. Whittaker who proposed a 5 kingdom classification, which consisted of Kingdom Monera, Kingdom Protista, Kingdom Fungi, Plantae, and Animalia. And he classified these, he came up with this 5 kingdom classification on the basis of various characteristic features like cell type, cell wall, nuclear membrane, body organization, mode of nutrition. And this is a very important table which we should revise again. So let us see what are the features of various kingdoms. So we find in case of cell type, only Monera has prokaryotic cells. The rest of all the kingdoms have eukaryotic cells. As far as the cell wall is concerned, we find that cell wall is completely absent in Animalia and is present in different forms in Monera, Protista, fungi, and Plantae. So in Monera, the cell wall is non-cellulosic, whereas in Plantae it is cellulosic. In fungi, the cell wall also contains chitin. As far as the nuclear membrane is concerned, we find that only Monera, as their cells are prokaryotic, the nuclear membrane is absent. Whereas all the rest of the kingdom's cells, since they are eukaryotic, the nuclear membrane is present in them. So what about the body organization? We find that Monera and Protista are unicellular. Fungi, Plantae, and Animalia are multicellular. Various cells form tissues and organs in Plantae as well as in Animalia. So in Animalia, even organ systems are produced. As far as the mode of nutrition is concerned, we find that fungi and Animalia are only heterotrophic, whereas Plantae is only autotrophic. In Monera and Protista, we find both autotrophic as well as heterotrophic mode of nutrition. So this table, please don't forget, you have to revise it again and again and remember it. So since now we are going to talk only of prokaryotic cells in this kingdom, in this unit, that is Kingdom Monera. So we will revise prokaryotic cell again. So a prokaryotic cell does not have nuclear membrane. No well-organized nucleus is present. The nucleochromatin material which is present is free in the cytoplasm in the form of circular shaped structures. And the cytoplasm of this particular prokaryotic cell does not have any organelles, any membrane bound organelles except for ribosome. So there is no mitochondria, there is no Golgi apparatus, there is no lysosomes, and so on and so forth. Only ribosome is present. The cell wall of the prokaryotic cells is also a bit different, and the cell wall is, however, very complex in nature. And mostly the prokaryotic cells, they reproduce by asexual mode of reproduction. So let us see a kingdom which has only prokaryotic organisms. So this is the Kingdom Monera. So thus among these five kingdoms, it is this kingdom we are going to talk of. And we had also learned in the previous class that Kingdom Monera was also further subdivided into two categories, two domains, and hence a seven kingdom classification system also was developed. So what are these two domains in which the Kingdom Monera was categorized? That was Archaebacteria and Eubacteria. So let us see what is Kingdom Monera first. We will just see the introduction of Kingdom Monera. So Monera consists of bacteria, all the bacteria. I'm sure everybody knows what are bacteria. We talk about bacteria so often. We ourselves get so many bacterial infections. So bacteria are the sole members of this Kingdom Monera. And bacteria are most abundant microorganisms. You will find them everywhere. Even on our body, inside our body, there are some commensal bacteria present who do not cause any harm to our body unless and until we are unhealthy. If we are healthy enough, these organisms will not cause any harm to our body. As well as there are bacteria present everywhere around us, even in the soil and everywhere. So these bacteria can live everywhere. Some bacteria can also live in extreme habitats, very, very extreme habitats where no other organism can live, like in hot springs, in deserts, dry deserts, in snow, in deep oceans, etc. And they can survive in these particular extreme habitats. And many of these bacteria, they are parasites as well as saprophytes. But the bacteria overall show a varying mode of nutrition. So now these bacteria are divided into four categories based on their shape. So when we see a spherical bacteria, then it is called as a coccus. In plural, it is called as a coccus, and plurally we call it as cocci. If it is a rod-shaped bacteria, then we call it as a bacillus or bacilli. If it is a comma-shaped bacteria, we call it as a vibrium or vibrio. And if it is a spiral bacteria, we call it as a spirillum or spirilla. So this is a picture given in our textbook. And we find in the bacilla, there is even spore which is shown. So some bacilli or some bacteria produce spores, especially in unfavorable conditions. The bacterial structure is very simple, but they behave in a very complex manner. And as we have already seen, they have got a most extensive metabolic diversity. So we find that bacteria may be autotrophic or may be heterotrophic. If they are autotrophic, they may be photosynthetic, they may be chemosynthetic. We have already seen all these modes of nutrition earlier. And if they are heterotrophic, then they may be parasitic or they may be saprophytic. So let us begin with the first type of Monera, the first domain, that is Archaebacteria. Now, Archaebacteria, they are the most primitive bacteria. They are considered to be the oldest living organisms on the earth. And they live in some of the most harsh habitats. We have already seen the hot springs and so on and so forth. So they live in these harsh habitats where it is very difficult for other organisms to live. And hence they are also called as extremophiles. So they may live in extremely salty areas, like there are salty, very, very salty oceans like Dead Sea, which lies in between Jordan and the West Bank in the Middle East. And there are so many other lakes which are very, very salty, like Great Salt Lake of Utah, etc. So under in these salty environments, these bacteria live, which are called as halophiles. Then some bacteria live in hot springs. There are so many hot springs present. There are some hot springs in the world whose water is also acidic, like the Yellowstone National Park in US. So the bacteria which live there are called as thermoacidophiles. Then some bacteria live in marshy areas. What is a marsh? It is a land which is covered with water for a long period of time. So these bacteria which live in the marshy areas are called as methogens, and they actually act on the organic matter which is present in the marshy areas and convert or produce methane gas from it. So they are called as methogens. Archaebacteria differ from other bacteria in having a very different type of cell wall. So this cell wall is a very tough cell wall which helps this particular bacteria to survive in extreme conditions. So what does this cell wall of Archaebacteria contain? The cell wall of the Archaebacteria does not contain peptidoglycans as is found in other bacteria, that is in eubacteria. It contains a pseudopeptidoglycan, and it contains a chemical which is called as isoprene. This isoprene is a rubber-like material, okay, which is an ether-linked membrane lipids. And these are hydrophobic. They will not allow water or any harmful substance to enter inside the bacteria. So they will protect this bacteria from all the extreme conditions in which they flourish. So methogens are not only present in marshy areas, but methogens are also found in the guts of several ruminant animals. So everybody knows what are ruminant animals. They are the animals which can digest the fiber content of the plant food which they eat, which we cannot digest. Human beings cannot digest the fiber content. But these animals do digest the fiber content. As they eat the plant food, they chew it thoroughly in the form of cud, they swallow it, then they again get back, regurgitate that material into the mouth, again chew it, and again swallow it. In this way, they cause complete digestion of the fibrous material, the fiber content of the vegetable food material. So, cows, buffaloes, goats, sheep, all are ruminant animals. And these bacteria, which are present in their gut, are responsible for production of methane, a biogas, which is obtained from the dung of these particular animals. So this biogas can be used for electricity production, for production of heat. It can also be used as a cooking gas in the kitchens. So this was all about Archaebacteria. Now let us move on to eubacteria, which is the second domain in Kingdom Monera. So eubacteria can be divided into different types depending upon what is their mode of nutrition. So eubacteria may be a photosynthetic autotroph, it may be a chemosynthetic autotroph, or it may be a heterotroph. So let us see first, before we see the different types of eubacteria, we should know what is eubacteria. So eu means true. So eubacteria are actually true bacteria, which are found everywhere on the surface of the earth. So the archaebacteria are found only in extreme conditions, but eubacteria are found everywhere on the surface of the earth. And they are characterized by the presence of a rigid cell wall. And if they are motile, then they also have flagellum, a hair-like structure attached to it, which helps it to move. So this rigid cell wall, the cell membrane of this bacteria, contains peptidoglycans and also contains ester-linked phospholipids. So let us see first the photosynthetic eubacteria, autotroph. So this is a cyanobacteria or a blue-green algae. Is another name for the cyanobacteria. And you can identify cyanobacteria very easily in any stagnant water or polluted water which you see around you in the environment. And you find greenish hue to this particular water, a stagnant water. So these cyanobacteria have chlorophyll A, which is similar to green plants, and hence they are photosynthetic autotrophs. They are unicellular, they may be present singularly, or they may form colonies. The unicellular bacteria may come together to form colonies or clusters, or they may attach to each other to produce hair-like filamentous structures. They may be present in fresh water, they may be present in marine or seawater, or they may even be terrestrial. They may be present on the land. The colonies are generally surrounded by a gelatinous sheath around them. And many times they form blooms, that is, they multiply rapidly inside the water and they cause the water to turn green. So a polluted water body, very commonly we find it turns green because of a cyanobacterial bloom. Some of these cyanobacteria can fix atmospheric nitrogen in specialized cells called heterocytes. So you can see there is a diagram of cyanobacteria, which is given in the textbook. And we can find that there are cells which are arranged in the form of, attached to each other, arranged in the form of a thread-like filament, filamentous arrangement this is. And in between there are these heterocytes, which can fix the atmospheric nitrogen. And you can also find that the whole of this filament is covered with a gelatinous mucinous sheath around it. So, which are these bacteria which fix the atmospheric nitrogen? The examples are nostoc and anabina. And this nitrogen is fixed in heterocytes. What are these heterocytes? They are specialized nitrogen fixing cells which are formed during nitrogen starvation by these filamentous cyanobacteria. So this nitrogen is utilized for production of all proteins, proteinaceous organelles and parts of this cyanobacteria. They fix nitrogen from air using the enzyme nitrogenase. So this was about the photosynthetic eubacteria, that is cyanobacteria, which is an autotroph. Let us move on to other type of eubacteria, which is another autotroph, but it is a chemosynthetic autotrophs. So what do these chemosynthetic autotrophs do? They oxidize various inorganic substances such as nitrates, nitrites, and ammonia, and many other substances which are present in their habitats and generate energy or ATP by this particular process which they utilize. So you can see in this picture, there are some thermal vents which are under the ocean. And these chemosynthetic autotrophic bacteria very commonly are present in and around these thermal vents because many minerals are present in and around these thermal vents. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, sulfur, etc. So what are the various examples of chemosynthetic autotrophic bacteria? Eubacteria. They are nitrifying bacteria. which which are called as nitrosomonas, and they uh, oxidize various nitrogen containing compounds. Then sulfur bacteria, which are called as begiotoa, and they oxidize various sulfur containing chemicals, sulfur compounds. Hydrogen bacteria, methane bacteria, iron and manganese bacteria, carbon monoxide bacteria. So you see these chemosynthetic autotrophic U bacteria, they can oxidize so many minerals and inorganic substances and can generate energy in the form of ATP and utilize it. Now let us move on to heterotrophic eubacteria. So till now we have seen autotrophic eubacteria in the form of photosynthetic cyanobacteria, and we have also seen chemosynthetic autotrophic eubacteria. So let us now move on to heterotrophic eubacteria. So these heterotrophic eubacteria, they derive their energy from organic compounds. They are the most abundant bacteria in the nature, and majority are important decomposers in the nature. Many of them have significant impact on human affairs. So we find that there are so many bacteria which can be utilized in for various functions. For example, some bacteria help in making curd from the milk. They help in curdling of the milk. Some bacteria are utilized for production of antibiotics, which are drugs, therapeutic drugs, which are used against the bacterial infections. Then some bacteria can help in fixing nitrogen in the legume of the roots of the plants. Some bacteria are pathogens also, and they can cause diseases in human beings, in plants, leading to destruction of crops. Also, they can cause diseases on farm animals and pets. So some of the diseases, common diseases which are caused by the eubacteria are heterotrophic eubacteria are cholera, typhoid, tetanus in human beings, and citrus canker in the plants. So how do these eubacteria reproduce? How do these bacteria reproduce? Not only eubacteria, all the bacteria, they reproduce very commonly by fission, which is called as binary fission. And you can see in these diagrams that how a single bacteria is dividing, single cell of the bacteria is dividing into two cells. So the nuclear material which is present in the cytoplasm, as well as the cytoplasm and its few organelles, are dividing into two and getting distributed in the two daughter cells which are being produced from this particular single cell. So this is called as binary fission. Sometimes under unfavorable conditions, they also produce spores, which is also an asexual method of reproduction. They also reproduce by a sort of sexual reproduction. We have talked about it earlier also, by adapting a primitive type of DNA transfer from one bacterium to the other. We have seen that in sexual reproduction, what happens that the genetic material or DNA of two parents, they come together and produce an offspring of the same kind as that of the parents. So in the bacteria, some amount of DNA transfer may occur, which may look like sexual reproduction. So a bacterium may take up DNA which is present outside it by various methods like conjugation, transformation, and transduction, and may lead to reproduction. So bacteria may also show a sort of sexual reproduction. Lastly, let us see what is mycoplasma. Now, this is also a type of bacteria, but it has got some special characteristics. Like this mycoplasma completely lacks a cell wall, and they are the smallest living cells which are known to survive without oxygen. Many of the mycoplasma are also pathogenic, that means they can cause various diseases in plants as well as animals. So here we end with Kingdom Monera. So I hope you understood and liked the class. If you have understood it, then please share it with your friends and also subscribe me to them.